There are only a handful of drugs available to treat these fungal infections. There are really three main classes of antifungal drugs that are used. And there's a bigger problem in that resistance to some of these antifungals is growing and spreading throughout the globe.
Hello, everyone, and welcome back to People, Parasites, and Plagues, a podcast aimed at delivering information about the fascinating pathogens among us from the scientists who study them. I am David Peterson.
And I am Kim Klanowski, your host for today's episode. If DNA is the text of the instruction manual for life, then chromatin is the way that the manual is organized, complete with bookmarks, highlighted sections, and notes in the margins. Today's guest studies how cells use this organizational system to control which instructions get read and when.
Our guest today is Dr. Zachary Lewis, Associate Professor in the Department of Microbiology. Zach, welcome to the podcast.
Thanks for having me.
Well, welcome. The first question that we like to ask our guests is about their pathway to science. So can you tell us how you were first interested in science, starting wherever you choose to start?
Sure. I guess I will start at the beginning of my undergrad career. So I think, like many undergrads, I came to college expecting to become a doctor, and I was a pre-med major. I took my freshman chemistry classes, and then I went home to where my parents lived at the time in rural South Georgia, and I worked for a summer in Seminole County Hospital, which is in a really small town. And I decided I didn't particularly like being around elderly sick people very much, and that perhaps medicine was not the dream job that I thought it was going to be. So I came back to Athens. I didn't really know what I was going to do. And I kind of randomly got a job working in a plant pathology lab. And that's where I kind of fell in love with being in the lab and doing science. So I guess I worked in Dick Hussey's lab in the plant pathology department. My job was to basically go out to the greenhouses on Riverbend Road once a week and inoculate tomato plants with these juvenile root knot nematodes. They would burrow into the plant roots, and I would then collect the plants that I had inoculated 3 or 4 weeks before and bring them back to the lab and spend the rest of the week looking through a microscope, dissecting adult nematodes out of the plant roots. And they were fascinating. So, these pathogens have these little straw type structures in their mouths, and they burrow into the plant root, and they would inject this straw into the plant cell and secrete a bunch of factors that would transform the cell, cause it to enlarge and produce lots of cytoplasm, and it would slurp up the cytoplasm like it was at 7-Eleven drinking a Slurpee, and I thought it was just the coolest thing ever. So eventually one of the postdocs in the lab figured out that I enjoyed this kind of thing, and he started teaching me some molecular biology, working with DNA, doing some protein purifications, and I was hooked.
So I just want to go to this because I'm a gardener, and I know that this is like root knot nematodes are like one of the main things that gardeners, especially people who plant tomatoes, are like, no, because supposedly once it gets in the soil around the area, like you can't really grow there anymore until you treat it, because you would just get it over and over and over again. And you get no production. So It's probably funny if anyone who is a gardener is listening to this thinking like people actually study root knot nematode.
Yeah, I mean, this was a huge program at UGA. They were breeding for resistance. They were also, trying to understand mechanisms of pathogenesis so they could develop anti-nematode agents to try to treat these infections. But yeah, it's a huge problem both in people's home garden, but also in large-scale commercial agriculture.
So while you were an undergrad there in plant pathology, did you get exposed to pathogenic fungi? Because I know there's a lot of fungi that are pathogenic to plants.
Yeah, there are. I didn't. So I was a biology major here at UGA, and I don't think I studied any pathogenic fungi as a biology major. After I started working in this nematode lab, I looked for other opportunities to work in labs, and I ended up working with Claiborne Glover, who was a faculty member in the biochemistry department. And Claiborne used yeast to study casein kinase 2. So I started working with yeast as a model system, and that was where I first kind of became interested in fungi, but not necessarily from the pathogen side of things, more fungi as a model system.
So actually my first introduction to lab work as an undergrad was working on Penicillium chrysogenum, doing protein purification. And I loved it. I didn't love the enzyme kinetics part of it, but I love the whole working in the lab, figuring stuff out. And so fungi started me on my career.
Yeah, excellent.
So Zach, did you stay at UGA or did you just find your way back here? I
just randomly found my way back here. So I went to graduate school at Texas A&M, also in the biology department. And I had become interested in the genetics of behavior. And they had a circadian rhythm program at Texas A&M University that I was drawn to. When I went there, I really wanted to work on cyanobacteria, which have a circadian clock that regulates photosynthesis and nitrogen fixation to different times of day. But when I arrived, there was a new faculty member that worked on circadian clocks using fungi as a model system. And I really liked the idea of having a genetically tractable microbial system that was a eukaryote to study the mechanisms by which organisms tell time. That's when I first really got into fungi, was as a graduate student, and I worked with a filamentous fungus to study circadian clocks.
So, and after your postdoc, well, that was your grad school, so I assume you did a postdoc.
Yeah, so in order to study circadian clocks in fungi, we used to monitor their developmental rhythm. So they would produce spores once a day. You can culture these fungi in the dark, and their internal clock would cause them to develop spores once a day. And then you could measure that. It was basically a fossil record of their internal biological rhythm, because they make these spores in growing rings. Mm-hmm. And that was fun, but it was a lot of work. So we were trying to develop a reporter by integrating firefly luciferase into the genome under control of a circadian clock-controlled promoter. And I would introduce this into the fungus, and I would get beautiful circadian rhythms of luciferase expression for about a week, and then the fungus would never express this gene again. And I eventually learned that it was being epigenetically silenced. The fungus did not like that we put a firefly gene into its genome. It was able to recognize it as foreign and repress it. So I got really interested in how cells use chromatin-based mechanisms to regulate gene expression. And so I went to University of Oregon to do my postdoc in that area.
Right. And that kind of leads us into what's happening today. It was a perfect segue. So, Zach, do you want to talk to us about what you're doing today in regards to chromatin structure and gene regulation?
And maybe if you can start out with just giving us a little definition of what chromatin is. Sure.
Okay. So in my lab, we do three pretty different things. Mm-hmm. So most of the lab works on chromatin structure and gene regulation. And so essentially this is studying how the packaging of DNA inside the nucleus influences gene regulation. So DNA, as you guys know, gets wrapped around histone proteins to form these arrays of nucleosomes. If you look at this under an electron micrograph, it would be, or under an electron microscope, these would look like beads on a string. Mm-hmm. And these beads on a string can then fold and assemble into higher order structures that can be highly condensed, in which the DNA becomes very inaccessible, or they can be more loosely assembled, and that DNA is accessible to transcriptional machinery. So the level of packaging can determine whether a gene is active and expressed or silent and repressed.
I also like to, I know the bead on the string thing is a great analogy, but the other thing is like a ball of yarn, right? Like, so if you're a knitter or something and you have your yarn just laying around in a big messy pile, right, everything's accessible. All the parts of it are. But when you have it in that ball, when you wind it up at the end of the day of knitting, it's so tight, right? You just can't get to some of those areas. So.
Yeah, exactly.
There we go.
Yeah.
Perfect analogy.
I like the ball of yarn analogy. Every once in a while, when we go to an epigenetics conference, there's a nice, colorful ball of yarn on the front of the program.
I love it. I love it. So that packaging and the kind of the tightness of the DNA alone is going to influence gene expression, but there's other things that are going to influence gene expression, right?
Yes, exactly. So packaging is one aspect of chromatin structure. Another aspect of chromatin structure is the deposition of small molecular modifications that can occur on the packaging proteins or on the DNA themselves. So these are things like methyl groups or acetyl groups that can be covalently attached to DNA or histones. And those modifications act like signals that can either recruit transcription machinery or transcriptional activators, or they can recruit repressors that make sure genes are stably turned off. So we work with one of these histone modifying enzymes. It's a complex called polycomb repressive complex 2, and it puts a methyl group on a single lysine in histone H3, and through that histone modification, it leads to very stable and heritable gene repression. So that modification is critical for multicellular development, and it's frequently misregulated in disease states such as cancer.
So there must be a lot of genes that have that particular histone in them. How does the enzyme know which one to modify or not? Is it like across the whole genome? It'll turn lay down a methyl group and turn things on or off, or is it gene by gene?
Yeah, it's a good question. It tends to be gene by gene. In early development, this methyltransferase can be recruited to specific genes. It can also be co-recruited along with activators in a stem cell, for example. So that gene has the potential to be expressed or repressed depending on which cell lineage that stem cell adopts. And so if it adopts lineage B, the gene may be repressed by the methyl modification. So it tends to be recruited to specific genes by DNA binding proteins.
They're probably very specifically regulated during development too, right? So there's probably patterns and things that are preserved, right? Which is a kind of a different level of regulation.
Right. Once a cell adopts a lineage and the patterns of methylation are laid down in that particular cell type, one interesting thing about this chromatin regulator is it is self-propagating. So once the repressed or methylated chromatin state has formed, it can read that repressed state and then reproduce it every cell division. So it can preserve a repressed gene state at a specific site for the lifetime of an organism.
Now, I would assume that pattern would reset in a spore. Is that correct? Because it's kind of restarting the developmental program, or is that not the case?
It's a good question. It's one of the things that we're working on. So in mammals, this is pretty well worked out that it resets in the early embryo. But in fungi, we don't really know when it resets. We do know that it regulates cell differentiation in a way that's very similar to what happens in mammals or plants, but we don't quite understand the signals in fungi as well as we do in some other systems.
So what happens with cancer? Is it the enzyme that's dysregulated? Is it the way that it's putting down these modifications? Is it, this kind of...
Both of those things can happen. It depends on the particular type of cancer. So approximately 80% of pediatric gliomas are caused by a mutation in the histone amino acid that is methylated. And in humans, we have 11 gene copies of histone H3, but mutation of only one of those will actually bind to the methyltransferase and trap it, which essentially acts as a dominant negative, and it's not able to methylate its normal targets. It's not able to repress those normal targets, and that mutation is causative for these gliomas.
So that particular histone methyltransferase is kind of evolutionarily conserved. Are some of its target genes also conserved?
So,
I mean, we're talking about quite a stretch of evolutionary time there, but
right. I mean, fungi and animals, I think, diverged approximately 1 billion years ago. So the target genes in fungi don't look the same as the target genes in animals, but within the animal lineage, for example, it methylates or represses some of the same target genes in a fruit fly as it does in a mammal.
So I guess that particular pediatric cancer you mentioned, that could be screened for, and I don't know if they actually do screening for it. Is it?
As I understand it, most of these are somatic mutations, so they're not inherited mutations. They occur during the patient's development, and this mutation is acquired during development and gives rise to cancer. But there are other mutations in other types of cancer that impact this enzyme. So in certain melanomas, there's a mutation in the methyltransferase that causes it to become hyperactive, and that's associated with a fairly aggressive subtype of melanoma. So the level of activity is critical. If you have too little, this can lead to aberrant gene activation and drive tumor development. And if you have hyperactive methylation, this can lead to aberrant silencing of tumor suppressor genes, which can lead to cancer through a different mechanism.
So there's a sweet spot you want.
Yes. It needs to be performing with the right level of activity at the right locations in the genome.
Yeah, so there's a lot of things that can go wrong.
So do you work on any histone acetylating enzymes as well?
We do work on acetylating enzymes. So we work on one that's surprisingly linked to repressions. Typically, acetylation is thought of as a modification that turns on genes or activates genes. But I think a lot of these generalizations don't turn out to be true the deeper you dig. So we work on one acetyltransferase that's really important during DNA replication. And when you disrupt that acetyltransferase, it also has a silencing defect.
So how are you using some of your studies in the plant biology world?
So we haven't done a whole lot of work on plant biology. For our chromatin work, we use a model fungus called Neurospora crassa. And Neurospora doesn't bother anybody. It's a soil saprophyte. It doesn't infect plants. It doesn't infect people, but it's been used as a genetic model system for 100 years. There are lots of tools. And what we learn in Neurospora, I think we can apply to other fungi. So one thing that's interesting about the target genes of this methyltransferase is that they do kind of vary depending on which fungi we look at. So if we look at certain plant pathogens, a number of their effector genes that mediate plant pathogenesis are repressed by this methyltransferase, and then those genes are held in the off state until plant infection is well underway, and then these genes are coordinately activated. So it provides a mechanism in which a large number of genes can be regulated in a coordinated way to, make a major cell type switch. So from growing in the soil to growing inside of a plant, for example. Some of what we learn in Neurospora, I think, is relevant to what's happening in these plant pathogens, but we haven't done a whole lot of work on those plant pathogens ourselves.
So one other thing that I think some of your research touches on is the role that chromatin plays in genome stability.
Yeah, so most of us think about the genome coding for genes, and that being the most important thing that the genome does, but there are large regions of the genome that don't code for genes, but instead they form important structural parts of chromosomes. I think the most obvious example is the centromere, which serves as the site of kinetochore attachment, and that facilitates, the separation of chromosomes during cell division. And those regions of the chromosome are highly repetitive. They tend to lack genes, and they tend to be very large. So those regions of the genome are packaged with a different specialized form of histone methylation, And if that methylation is defective, then those regions can frequently exhibit double-strand breaks. The segregation of chromosomes during cell division tends to be error-prone if chromatin isn't formed properly at those sites of the genome. So we've done a little bit of work on that as well.
That's a pretty profound effect. I mean, whether you're talking double-stranded breaks or not segregating chromosomes properly, that's almost a game over sort of situation.
Yeah, not surprisingly, the mutations in those factors that regulate that process tend to be lethal. It's a fundamental mechanism for preserving genetic information from one cell to the next.
Zach, one of the things I know about fungal infections, and I'm thinking mostly of human, but treating them can be problematic. It seems like there's not a lot of good drugs and there's resistance, and maybe you can lead off talking a bit about that.
So beyond our work with Neurospora as a model fungus, we do work with a number of human fungal pathogens, and we work with those in two different ways. One, we study chromatin structure and how it relates to antifungal drug resistance. And then our other work is focused on developing antifungal therapeutics that are more effective and less toxic. But human fungal infections are a growing problem. I think for a long time, we haven't appreciated how big of a disease burden there was due to fungal infections, partly because they present like lots of other fungal infections. You know, a Aspergillus lung infection presents a lot like bacterial pneumonia. And so most, if a patient dies from pneumonia, it doesn't always get classified as a fungal pneumonia or a bacterial pneumonia. But, current estimates suggest that there are over 6 million invasive fungal infections a year globally, and over 3 million deaths attributed to these fungal infections. So it's a significant problem. There are only a handful of drugs available to treat these fungal infections. There are really 3 main classes of antifungal drugs that are used. And there's a bigger problem in that resistance to some of these antifungals is growing and spreading throughout the globe.
And there's a lot of bad side effects to these drugs too, right?
Yeah, even, the best antifungal drug that we have is a molecule called amphotericin B. Mm-hmm. It was first used in the 1950s. It's still in use, but it's highly toxic to patients.
That's the one I think physicians refer to as amphoterrible.
Mm-hmm. Yeah, amphoterrible. I had a clinician one time tell me they call it shake and bake because the patients go through cycles of fever and chills as soon as they are put on this medication. So it's a highly effective antifungal, but it's pretty toxic to patients.
It's kind of amazing that we're talking about in the 50s and we haven't really made a lot of progress in the antifungals. It seems incredibly slow. So it must be pretty tough.
I think it's challenging. I think it's also the same problem that plagues antibiotic development for bacterial infections. It's not especially profitable for drug companies to develop new antifungal therapies. So the pipeline has lagged.
Yeah, because oftentimes, I guess what pharmaceutical companies are worried about is they come out with a new hot, antifungal, antibacterial, antiparasitic, and within 4 or 5 years there's resistance. And so they don't have, although amphotericin has been on the market, as we said, for quite a while, but that's not true for all of these.
Yeah. And amphotericin, fortunately, doesn't have a huge resistance problem. I mean, there are some fungi that are moderately resistant to amphotericin. For Aspergillus, which is a fungus that we work with probably more than any of the other fungal pathogens, the frontline clinical drug is an azole class drug. It inhibits production of ergosterol, which is a component of fungal membranes. Fungi don't make cholesterol, so they make this similar molecule called ergosterol. And the azole drugs inhibit one of the biosynthetic enzymes that is in the ergosterol production pathway. It's a frontline drug in the clinic for pulmonary aspergillosis and other fungal infections, but it's also widely used in agriculture. So your strawberries that you buy in the supermarket are coated in azole fungicides. And it's pretty much appreciated at this point that the predominant strains that show up in the clinic were selected for drug resistance by Dutch tulip farmers. They spray loads of azole drugs on tulips. This gave rise. Aspergillus is not a pathogen of tulips, but it lives in the soil. This selected for drug-resistant Aspergillus fumigatus, which has spread around the world. It occasionally ends up in patient lungs, and they're not treatable with our most effective and least toxic front.
So there's no regulation to control this, really. I guess I'm assuming it's, I don't want to blame the Dutch, but I'm good.
No, I mean, this is a worldwide problem, right. And it's not restricted to the azole class of antifungals. There are additional antifungals being developed that are still being developed in parallel for use in the field and for use in the clinic. You know, there are a couple folks at UGA that work on this. Michelle Momini and Maren Brewer have spent a lot of time working on this. And
I think we need to get to the Netherlands for our next episode and investigate this more thoroughly.
I do know it's a big problem, and here's why I know. I mean, I alluded to the gardening, my love of gardening in the beginning of this episode. So growing tulips in Georgia, you can't, we don't get cold enough, so you don't have a return of your tulips. So I buy my tulips every year, and I have to pre-chill them for like 90 days or something like that before I put them in the ground, so I can give them their fake cold in Georgia to kind of get them into that right budding time. And I mean, you can see the fungus. I mean, I get them, I put them in my refrigerator, and you can start to see the fungus emerging on the bulb. So they've got to be covered in it. You know, I keep them in, I have a dedicated refrigerator and all of that stuff. So don't worry about my food having fungus all over it. But the point is like, I get their problem, right? But you know, sometimes when you swing that pendulum too far, there's repercussions, right? And that's clearly what we're seeing.
So the actual mutation that gives rise to approximately 80% of resistance in the clinic is a small duplication of DNA in the promoter of the drug target. So it contains 2 transcription factor binding sites, and just duplicating 34 bases of DNA is sufficient to increase expression of the target so that it's no longer sensitive to the azole drug.
That's a pretty elegant solution.
And so,
it is.
So part of our work is trying to understand the mechanism of resistance that results from this duplication.
So I know that you also work on another approach to specifically targeting fungus with drug-loaded liposomes, and you've got a cool name for it, but I'll let you talk about that.
Yeah. So this is the last project that we work on in the lab, and it's been a really fun collaboration with 2 other investigators at UGA, Rich Meager, who's in the Department of Genetics, who's really the leader of this project. Rich is a force of nature. And then Xiaorong Lin, who's a faculty member in my department in microbiology. And years ago, it occurred to us that we could potentially improve the efficacy of antifungal drugs just by targeting them to the fungal cells. And we knew that amphotericin B, which is this toxic but highly effective antifungal, is typically delivered to patients as a lipid nanoparticle. So they call them ambosomes, and they are 100 nanometer little lipid particles that are loaded with this antifungal drug. And that liposome packaging increases the circulation time in the blood, and it enables better tissue penetration, and it reduces toxicity to patients compared to the non-liposomal drug. But it's still a pretty toxic formulation. So it occurred to us that if we could just point these liposomes directly to the infection site, we might be able to improve the efficacy of the drug while at the same time reducing the dose that was needed and hopefully reduce toxicity to the patient. And so to do that, we've coated the liposomes with mammalian immune receptors that bind to sugars on the surface of fungal cells, and it tends to work great.
Kim just got really interested in this now.
I am. I'm very interested in this.
Yeah, so we used the dectin receptors that are carbohydrate binding receptors There are pathogen receptors on the surface of mammalian immune cells. And we've made a number of different versions of these now. We started with Dectin-1, which is a protein that binds to a beta-glucan polymer, and fungal cell walls are coated in beta-glucans. So these are receptors that our immune system uses to recognize fungi where they're not supposed to be.
It is funny that you bring this up now, because when we teach immunology, like, we just kind of casually mention these receptors, like very casually. Like, maybe there's clearly an important role and we just don't spend enough time talking about it.
Yeah, I mean, I think that they do a good job of keeping fungi out of places they're not supposed to be. Our skin is covered with fungi, so generally they're not pathogenic.
I think that's the reason why, right? Is for most immunocompetent people, we have all the appropriate immune mechanisms to deal with fungi for the most part. Yeah. So, to be able to really study fungi responses in humans, you have to usually immunosuppress to be able to study what is actually happening. You know what I mean? So it makes it, that's why we kind of casually gloss over it. We have these things and they keep the...
Yeah, it's an important point, right? Because most of us are not at high risk of experiencing or developing one of these life-threatening fungal infections. It's really immune-compromised patients that are at risk. But with modern medicine, that group of patients is growing. So patients with HIV are at high risk, but cancer patients that are undergoing stem cell therapies,
aged patients,
and patients that are taking immunosuppressant drugs for autoimmune disorders are all at high risk for developing these infections.
Yeah, that's definitely growing. So, Zach, what do you like to do in your free time when you're not thinking about fungi or?
He's always thinking about fungi.
Yeah, I only think about fungi. I like living in Georgia. So I very randomly ended up back in Georgia. I did my postdoc in Oregon, and when I was on the job market, UGA was hiring 3 faculty members that worked on fungi. I think it was the only job search that was for fungi in the whole country. So I ended up back in North Georgia, but I love it here. So my hobby is I like to go camping. I like to go to North Georgia and camp and hike in the mountains. I like to go to the coast and camp on the barrier islands. Sometimes I go to Florida, but not too far into Florida. But camp in the Panhandle. But that's my major hobby, is to get out of town.
That sounds like a good way to unwind. It really does.
Up.
Yeah, I like to find places where there's no cell service or Wi-Fi, so I don't have to answer any emails for a couple days.
Yeah, that actually sounds really good.
When I go out of town on vacation, I usually tell my department head, I hope he doesn't listen to this podcast now, but I usually tell him, where I'm going to be, there's just not good cell service, so it's really hit or miss.
Yeah, I can recommend some places where that will be completely true.
Yeah. Well, I would like to know what those places are. We'll talk off air.
So our guest today has been Dr. Zach Lewis. Zach, it's been great having you on the podcast.
Thanks for having me.
So links to additional information about Dr. Lewis's research can be found on our Instagram account, which is at PPP Podcast UGA.
As always, we appreciate you tuning in today. And if this is your first time listening to the People, Parasites, and Plagues podcast, we encourage you to check out the other exciting episodes launched last season. Please rate the podcast, and if you have any questions or suggestions for future episodes, reach out to us via email at ppp@uga.edu. This podcast is brought to you by the Faculty of Infectious Diseases and the Grady College of Journalism at the University of Georgia. It is supported by the University of Georgia through the Office of Research, the UGA Graduate School, and the College of Veterinary Medicine. Special thanks to our production assistant, Justin Nemitz, for research, editing, scheduling, and keeping us on track.